Hello, and welcome back. This is Professor Young, and this is the start of Chapter 2, Biochemistry. And this is going to be a rather lengthy look into really what's going to become the foundation of physiology. It's going to become the basis of everything that we're going to be learning about and discussing really from here on out. And so, with that said, since there is a lot of material here, why don't we go ahead and jump right into this? By the way, this is going to be part 1 of 2. There will be a second WIMBA session that will be devoted strictly to macromolecules and how they relate to both biochemistry and the anatomy and function and all that kind of good stuff. So I guess what I would like to do is start out with a basic review of the structure of an atom. We know that within the atom, we have 3 main subatomic particles. We've got our electrons, we have protons, and we also have neutrons. And really the difference between these 3 is that the electrons contain negative charges, and also that the electrons are found orbiting the outer shells. And so that surrounding the core of an atom, what we refer to as the nucleus, which would be located right here, that being the core or the nucleus, surrounding that are going to be these orbitals or these shells by which the electrons are going to pass. And you can see that we have an electron here, and we also have an electron there, and they are surrounding the nucleus. The neutrons, on the other hand, contain a neutral charge. They are found within the nucleus. And if we alter the number of neutrons within that nucleus, we simply change that number, and we create, trying to type and talk at the same time, it doesn't work. We create what we refer to as an isotope. And of course, if we change the number of electrons, we create ions by changing the number of electrons. And then, of course, we have protons. And protons exist, again, within the nucleus. And they contain a positive charge. And keep in mind that that positive charge is going to interact directly with those electrons and their negative charge. It's kind of the glue that's going to hold these atoms all together. And again, if we alter the number of protons within the nucleus, altering number of protons, it's going to change the atom. It's going to create an entirely new atom for us. The number of protons normally are going to equal the number of electrons within an atom. When it doesn't, then you kind of know that we've entered into that realm or that area of creating ions. And so if we have more electrons or fewer electrons, then we know that that number has been changed in some way. It's either given up electrons or it's accepted electrons, and it's formed into that ion. Typically, an atom that has not interacted with another atom in any way, shape, or form, the number of protons are going to equal the number of electrons. I also want you to remember that the electrons that are going to be involved directly in bonding are going to be valence electrons. Again, valence electrons are the electrons found in the outermost shell. And they're going to be the electrons that are going to be involved in bonding. They're going to be the electrons that are going to be involved in bonding. So the valence electrons are the electrons that are going to be found in the outermost orbit, furthest away from the nucleus, and there are going to be the electrons specifically that are going to be designated within the reactions for bonding. And if you think about it, that kind of makes sense. If the electrons are further away from the nucleus, there's going to be less attraction to those electrons. Right? What holds this entire system together is the fact that you've got a positively charged nucleus with these negatively charged electrons floating around. We know that opposites are going to attract. There's going to be a pull that's going to be tugging those electrons in to stay within the orbit of that element. Well, the further away from the nucleus you become, the less that pull is going to have an impact. The less attraction there's going to be between that positively charged nucleus and those electrons on the outer orbit. And so those are the electrons that are going to be more easily or readily available for bonding. That's what we refer to as the valence electrons. And so this here really, this here becomes a critical group of electrons for us to understand. In fact, when we talk about bonding, it's only those valence electrons, the electrons that are quote-unquote available on the outer orbit, that will be involved in bonding. Now, not to oversimplify this, I want to be careful that we don't do that. Keep in mind that we do have multiple shells. We do have multiple orbits by which these electrons are following, moving along. Electrons have the ability to move between the shells. At any one given point in time, we know that the first shell is only going to have 2 electrons. The second shell is only going to have 8 electrons. The third shell is only going to have 8 electrons. We understand that, but I also want to make sure that it's clear to understand that the electrons can move between those shells. And so there's a constant movement of electrons. As electrons gain energy, they move further away. As they lose energy, they get pulled back in. And they gain energy and they move further away, and they lose energy and they come further back in. It's all about whether or not they can withstand or overcome that attractional pull, and to what degree can they overcome it. The elements that we are going to be most concerned with are the elements that you see here, these 6. And in lecture, I did add 2 more. I believe I added potassium, which has a K for a symbol, and we also added oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus, potassium. Why don't I remember what the other one was that we added? Let's see here. I don't remember. When it comes to me, I'll probably jump back in with it. But there are 8 key elements, and for some reason, the other one is sodium. Knew it would come to me. Sodium is the other one that we added at the last minute there. These really are the 8 probably most critical elements within the body. The first 6 make up the bulk of the body mass. Potassium and sodium kind of round out that group. as far as what's really involved and what's really critical for proper functioning within the cells. And so that's your critical eight right there: oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus, potassium, and sodium. Whenever we deal with almost anything in physiology, either one or a multitude of these guys are going to be involved in one way or another. Kind of looking at major ideas within the biochemistry of the body. One of the key groups of biochemical reactions or substances that we have are this concept of electrolytes, right? Salts. And these salts behave very purposefully within water. They ionize. They become charged. When we talk about ionization, right, what we are really saying here is that we have created ions. Right? That's what we're referring to when we talk about ionizing something. We are creating ions. How do we create those ions? Well, on the next slide, we're going to take a look at these common salts. But let's just, for argument's sake, let's take sodium chloride as an example. Sodium chloride is a typical salt. It's made of sodium and chlorine. When we put sodium chloride in water, it disassociates. It goes through disassociation. Right? When something disassociates, it comes apart. It breaks into multiple pieces. And so what's going to do the disassociation of sodium chloride in this case is water. Water is going to have the ability to pull apart the sodium and the chlorine. What does that, what powers that, we will talk about here in a couple minutes. We're not going to get into it right now. But understand that when we break apart that sodium chloride, we're creating ions. Specifically, we are creating Na plus and Cl minus. We're creating sodium that has more protons than electrons. That's what that little plus means. That plus means it has one more proton than electron. And we're creating chlorine, which now has one more electron than what it does protons. And remember, we're going to be giving up or adding electrons. Right? The form which that comes into play is going to be dictated by how the electrons are moving. In this case, sodium is giving up an electron, and chlorine is accepting of the electron. When the salt ionizes, when it disassociates and comes back into its basic principles, its basic parts, sodium ions, chlorine ions, that solution that is created is now capable of conducting electricity. It's capable of conducting electrical currents. And that's powerful. Everything in your body runs off of conductivity. The way we send messages from the nervous system to everything else within the body is based upon, founded on this concept that our body can transmit electrical impulses. And what's going to help promote that electrical conductivity is going to be electrolytes. That's why they're in our sports drinks, to help maximize the efficiency that our muscles are working. Our muscles work based off of electrical impulses through which the nervous system communicates. And so chemical reactivity, it's going to help our chemical reactions to respond and happen. Whether or not you're too dehydrated, too many electrolytes, you're going to dehydrate. Too few electrolytes, you're going to short circuit. And so the balance of our electrolytes are definitely going to impact the way that our water is moving throughout the body. And as we've already mentioned, electrical effects as well. It's one of the key reasons why at night, when you cramp up with a muscle cramp, Charlie horse, as we sometimes refer to them as, many of you already know you reach for a banana. Why? Because it's high in potassium ions. And potassium chloride is a salt. And so that potassium is going to help regulate electrical impulse. And we're going to learn about exactly how that happens. But it all comes back into this concept of electrolytes. Talked about the major electrolytes. I said that we would go ahead and look at this real quick. This is a very brief listing of the 6 top electrolytes found in the body. Calcium chloride. When it breaks down and dissociates into its basic ionized components or compartments, we have calcium ions and chlorine ions. Right? Disodium phosphate. 2 sodiums for each phosphate group. Right? That's a salt. Right? The ion specifically it's ionizing into is sodium and the phosphate group. Right? Magnesium chloride, magnesium ions, chlorine ions, potassium chloride, sodium bicarbonate, and sodium chloride. These are all things that we're going to come back to and touch on throughout the semester. Maybe not directly looking at these from the perspective of me saying calcium chloride or potassium chloride or sodium chloride, but we're definitely going to be utilizing and learning about how these ions help to elicit electrical responses and how these substances, how these ions play a role in maintaining osmotic balance. Free radicals are another hot topic issue that you see a lot of times whenever people talk about holistic health responses and how to go ahead and prevent aging and all that kind of good stuff. What a free radical basically comes down to is being a charged, an overly charged oxygen atom, primarily. We kind of usually define this as being an O negative, but that O negative is signified by that little dot, and that dot above that negative sign means that it has too many. It has an odd number of electrons within it, and it supercharges that oxygen molecule. What produces this free radical? Is normal metabolic reactions. You're producing free radicals right now. Right? The difference is you're producing them at a rate at which the body can go ahead and break them down. SOD, superoxide dismutase, is a naturally produced antioxidant within your body that goes ahead and neutralizes those extra electrons and really makes that oxygen molecule harmless by that point in time. Where free radicals really become dangerous is when we start looking at things like UV radiation, when we start looking at chemicals that we're putting within our body or that we're exposing our body to. Radiation, UV radiation, and various types of chemicals, cleaning agents, gardening agents, or illegal drugs, so on and so forth, have the ability to speed up normal metabolic reactions. And when you do that, you drastically increase the presence of the free radical. And free radicals causes tissue damage. It's why you age. And let me tell you something. You know, we constantly hear news reports about the harm in sun tanning and the harm in overexposure to the sun and the harm in overexposure to UV radiation. If I had my choice, which I do, I'm always going to choose natural sun UV radiation exposure over tanning beds, because even though it's a very short exposure within the tanning beds, it's a very high dose of UV radiation. And that's why people that over tan start to have that appearance of that very tough, leathery skin. It may look nice in the short term, but long term, you're doing such drastic damage to the skin cells themselves. And part of that aging process is being expedited. Why? Because of the high presence of free radicals. What's causing the free radicals? The high levels of exposure to the radiation, the UV radiation, that UV light. And the same thing with chemicals. And again, it could be cleaning solvents, it could be gardening fertilizers, it could be drugs, it could be any number of things. Cigarette smoke, alcohol, any of that stuff in high dose, it's going to cause damage to the tissue. And whenever you have to force a cell to increase the amount of energy that it does to repair itself, you are causing higher levels of free radicals. It's plain and simple. There's no other way that I can explain it. What do we use to fight them? We use antioxidants. It neutralizes it. It knocks off those extra odd number of electrons and brings everything right back into normal levels. And basically what it does is it knocks the electrons off and takes that oxygen and creates hydrogen peroxide. And then the hydrogen peroxide can then be broken down to release more oxygen, or actually it releases an oxygen and a water molecule. Right? Hydrogen peroxide, for those of you that don't know, it's H2O2. And so what, especially superoxide dismutase, what that does is it takes the free radical and combines it with the superoxide dismutase to create something called superoxide. And when it does that, it removes an oxygen. So now you've got oxygen that's being produced, or at least a half of oxygen molecule that's being produced, and what you have left over is what you've got left over here is you have a half of oxygen molecule and a molecule of H2O. And so you create water. And so you're neutralizing the free radical by using the SOD to basically create a half a molecule of oxygen, a half a molecule of oxygen and water, which is harmless. And our body can handle that a lot easier. Anions and cations. Anions are anything that are going to gain electrons. Cations are going to be anything that's going to lose an electron. And this is where those valence electrons are really going to come in play, because it's going to be those valence electrons that we're either going to be gaining or losing overall. And so if we're losing electrons, we're forming a cation, and it's going to have a positive charge. It's going to have a positive charge. Why a positive charge? Because we've lost electrons. Remember what I said in the beginning. The number of electrons and the number of protons are, for all intended purposes, equal. They're equal. When we create that cation, we've now lost an electron, which means the protons outnumber, and so that's why it gets that positive charge. If we're gaining an electron, then we're gaining an overall negative charge. Check out sodium and chloride as the example, and we spent some time in this in lecture as well. Sodium normally has not 10 electrons, but it normally has 11 electrons. And chlorine, on the other hand, doesn't normally have 18 electrons. It normally has 17 electrons. Well, what happens through the course of things as we are trying to stabilize the molecules, keep in mind, they want to have a full outer shell. They want to accomplish what we refer to as that octet rule. And the octet rule simply says that in order for a molecule or an atom, in order for an atom to be stabilized, the outer valence shell of electrons must be full. All right, now remember, that inner shell right here holds 2 electrons. This next shell holds 8 electrons. And this final shell that we're going to be concerned with also can hold 8 electrons. And so in order for that first shell to be full, all it needs is 2 electrons on it. In order for that second shell to be filled, all it needs is 8 electrons on it. In order for that third shell to be full and to meet the octet rule, it just needs 3 electrons on it. That's it. End of story. We're good to go. How do we get that in the case of sodium? If we look at sodium specifically, sodium has 11 electrons, which means it's got 2 on the first, right? Now it's got how many left over? It's got 9 left over, so that's enough to fill the second shell. It's got 8, but on that third shell, it only has 1 electron. How many does it want? It wants 8. So it's got a choice. It can either gain 7 more electrons, which, let's face it, that's a heck of a lot of work. Or it could give that electron away. Right? In the case of chlorine, on the other hand, has 17 electrons. And so it, again, is going to have 2 electrons on the first shell. Right? So now it's got 15 electrons left. It's going to have 8 electrons on the second shell. Now it's going to have 7 electrons left. And that means it's going to have 7 electrons on the third shell. It's not happy. It wants 1 more electron. It wants 1 more. Sodium has 1 that it's looking to give away. And so it's exactly what happens. Sodium says, I don't want this electron. Chlorine, do you want it? And chlorine says, yes, I want it. And it gives that 1 away there to the chlorine, so that chlorine now has 8. That outer shell, which is now empty because the sodium gave away 1 electron, drops down to the second shell, and it's now full. And so what ends up happening is the number of electrons in sodium drops down to 10. The number of electrons in chlorine jumps up to 18. Sodium gets a positive charge because it now has 11 protons compared to 10 electrons. Chlorine gets a negative charge because it now has 18 electrons to 17 protons. It's got 1 more electron than protons. And when we put this all together, we create essentially what we refer to as ionic bonding, right? The attraction between a cation and an anion. The kicker is, and there's always a kicker, the electrons are transferred. It's given away. It's giving it away. Sodium is giving its electron to chlorine with a catch. With a catch. Sodium is now bonded to chlorine. Sodium says, you can have my electron. I don't want it. I want to be stable. Chlorine, here is my electron. But I'm coming along for the ride. And that's exactly what happens. And when that happens, when one atom gives another atom its electron or electrons, we have what is known as ionic bonding. In its simplest form, we've got ionic bonding that is going on. Right? This is a relatively weak bond. It's a relatively weak bond. It's very easy to split up ionic bondings, especially in the presence of water. And we'll talk about that in a couple minutes. Opposite of ionic bonding is covalent bonding. Right? And with covalent bonding, we still have one atom giving another atom an electron, or two, or three, or four. But it's not a complete give and take. The difference here is, in a covalent bond, the electrons are shared. In other words, those electrons are going to spend some of the time with one atom and some of the time with the other atom. They're working together, sharing the electrons between the two atoms or the three atoms to maintain stability, to make sure that the outer shell, the valence electron shell, is full. That's something completely different than what is happening within ionic bonding. It is something that is completely different than what is happening in ionic bonding. And there are rules that dictate how the electrons are shared. There's rules that dictate how these electrons are shared. And the rule comes down to one word: electronegativity. That's the $1,000 word. That's the $1 million word right there. Electronegativity basically says whoever has the bigger nucleus is going to control the electron. The bigger the nucleus, the more protons are present. The more protons are present, the more positively charged that nucleus is. And the greater attraction or the greater pull it's going to have on that electron. If the atoms are the same size, you have equal sharing. If the atoms are not the same size, you are going to have unequal sharing. Same size, you have equal sharing. Differing size, non-equal sharing. We can take that and define it a step further and say that when we have equal sharing, we also have non-polar molecules. And when we have non-equal sharing, we have polar molecules. So when you have two atoms that are the same size or relatively the same size, it's going to be a non-polar substance. It's going to be equal sharing. Therefore, no one has more control over the electrons than the other one. We say that it's non-polar. And I'll explain what that means on the next slide. But if we have two atoms that are vastly different in size, then the nucleus on one atom is going to have a greater tug, a greater pull, a greater attraction to those electrons that are being shared, and it's going to hoard them more of the time for themselves. That's going to create non-equal sharing. And it's also going to create polar molecules. what we mean by polar here is that one side is going to be more negatively charged because it's going to have control of the electrons more, and the other side is going to be more positively charged. Why? Because it doesn't have the electrons most of the time. Let's take a look at that. Water is the great equalizer here. Water is the great equalizer. This, for those of you who do not know, is water. And water is made up of H2O, which basically means that we have oxygen here, hydrogen here, and hydrogen there. We have one oxygen bound to two hydrogens. And what we say is that the oxygen, because it is bigger, it actually has more protons in the nucleus. Because the oxygen has more protons in the nucleus, it's going to want to control the electrons that are being shared by little old hydrogen. It's going to want control over those electrons. And because those electrons spend more time with oxygen, we say it is partially negative. Now, I did a little fancy little symbol in class. I'm not going to be able to do that fancy little symbol here. But I can definitely type out partially negative. And that's what our electron is. That's what our oxygen is. Our oxygen is partially negative. On the other hand, our hydrogens are going to be partially positive. Why? Because they have smaller nuclei, fewer protons, less gravitational pull on those electrons. Are those electrons that they're sharing with oxygen and that oxygen is sharing with hydrogen, are they going to spend some time with hydrogen? Yes. But it's not going to be equal. And so that unequal sharing of electrons between two unequal sized atoms are going to create polar ends. One being partially negative, the other one being partially positive. And it is here where the magic of dissolving solutes takes place. In order for water to be able to dissolve anything, because keep in mind, water is the universal solvent. If you want to dissolve something, use water. And the reason why you use water to dissolve things is because it's polarized. If you've got a substance that can be broken down into a positive charge and a negative charge, the positive charge on the thing that you're dissolving is going to be attracted to the oxygen. And the thing that is negatively charged is going to be attracted to the hydrogens, the positive end. It's why sodium chloride dissolves, that's horrendous, hold on one second, dissolves so easily. Because we know that sodium is going to have a positive charge on it. And that chlorine is going to have a negative charge on it when it creates an ion. The positively charged sodium is going to be yanked and pulled away from the chlorine by the negatively charged atom, oxygen. And the negatively charged chlorine is going to be yanked away from the sodium by the positively charged hydrogen ends. And when you yank apart the sodium and the chlorine, it's dissolved. It's disassociated. Remove the water, sodium chloride bond back together with that ionic bond. It is also the reason why oil doesn't mix with water. Oil's nonpolar. It doesn't have a positive and a negative end to it. Water has nothing to grab onto it with. Hopefully that makes a wee bit of sense in the greater scheme of things here. Hopefully. Now, we've talked a little bit about water being the universal solvent, and I want to kind of take this a step further and just kind of review this real quick as we go through here. Our body is made up of hundreds of, maybe even possibly thousands of various mixtures. Right? This joining of more than one thing together in order to form a solution. The basis of almost all of those mixtures and all of those solutions is water. Water is the key to chemical reactivity and stability within our bodies. And that's key for you to keep in mind. Water is the basis of almost all of our mixtures, such as blood. You have red blood cells, white blood cells. You've got some other particulates floating through there. You have fats floating through there. You have plasma flowing through there. What holds all that together is water. It's also the basis of most of our solutions as well. And the reason why water can do that is because water has so many various properties to it. Water can act as a solvent, as we've already talked about. When we talk about a solution, we know that we have two aspects of being a solution. We have for a solution, you have the solvent and you have the solute. The solvent is what is doing the dissolving. In this case, it would be water. The solute is what is being dissolved. It is what's being dissolved. Now, keep in mind, there are some additional terms here that you need to be very well aware of. The only way water is going to be able to dissolve anything is if it is a hydrophilic solute. What we mean by hydrophilic here is hydro meaning water, philic, philia means love, water loving. Right? Hydrophilic solutes are solutes that can be disassociated in water. They've got the potential of having a positive and a negatively charged end that can intermingle with the positive and the partially positive and partially negative ends or poles on the water molecule. If it doesn't have that ability, then it's not a hydrophilic solute. Instead, it's hydrophobic, such as our fats and oils. If we're dealing with, and actually, let me jump here real quick, because this image here really depicts that perfectly for us. Water is being that solvent right there. Right? We can see that sodium ion is positively charged. We can see in the middle of all of those hydrogens, all those little blue balls facing it there, that the chlorine is negatively charged. Look at the sodium, the oxygen, those big red balls right there are oriented facing that sodium. They're partially negative. Right? And the hydrogens, the blue circles there, are partially positively charged. They're going to be attracted to that negative chlorine. It pops those guys apart. And we've talked about that. We've talked about that at some cases ad nauseum. We look at cohesion and adhesion. We look at these terms of cohesion and adhesion. Water has the ability to stick to things that are like itself. Right? That's cohesion. Water will stick to itself. That makes it a pretty powerful solvent, the fact that it can adhere to itself. But it also has the ability, what's happening here? This is what's wrong. It also has the ability to stick to other things. It also has that ability to stick to other things. When we talk about chemical reactivity, and we're going to talk a lot more about this when we get into the pH stuff, which I think we're going to end up doing on the next session. But when we talk about chemical reactivity, what we are really looking at here is how does water initiate chemical reactions within a system. And there's different ways that it can do that. We can add water to a system, something that is called hydrolysis, and it'll actually release energy. We can remove water from a system, something that is known as dehydration synthesis, and we can create new molecules for energy storage. We can go ahead and we can add water to something and create more of a base or create more of an acid. And so the reactivity ability of water is widespread. And the last thing that I want to leave you with today is this concept of thermal stability. And thermal stability has to do with the fact that connecting each of those water molecules together are hydrogen bonds. Hydrogen bonds connect water molecules. And it's those hydrogen bonds where we have energy storage. In other words, as we add heat to the system that has water in it, that heat does not very quickly raise the temperature of the water because that thermal energy can be sucked into those hydrogen bonds and stored there. That's why it takes water so long to boil, because what you're doing is all that heat energy you're adding in there initially goes right to those hydrogen bonds. And until you overload with heat those hydrogen bonds, water isn't going to boil. The temperature is not going to change. It's going to be a very steadily rising, slow-moving process. And so water has the ability to resist thermal change. Thank goodness. Because otherwise, your bodies would be overheating right now. Your body's creating heat left and right, and that heat needs to be dissipated out of the system. Well, before it can be dissipated out of the system, it's got to travel through the body. And it's got to be stored. And it's stored within the water molecules and gotten rid of, whether it's through sweat, whether it's through water vapor as you exhale while your breath is so hot, whether it's through the urine, whatever the means of dissipating that heat is, it's got to travel through water as the medium. If it didn't travel through that water, you would burn up your tissues. And if you burned up your tissues and damaged your cells, you wouldn't be able to function worth anything. And so that's really critical here as well. This whole concept here, all right, of thermal energy and stability. What I would like to do, I think, is stop here. I was going to continue a little bit more, but I think I'm going to stop here and save our discussion for acid and base chemistry for the next session. I'm going to let you kind of digest all of this kind of good stuff here that we've looked at so far. As always, if you have any questions, jot them down. Feel free to ask them at the beginning or at any point in time during our next lecture session, or shoot me an email. And I'll be sure to get back to you with an answer on your question. And so for now, this is Professor Young. This is the end of our look at Chapter 2, Part 1 of Biochemistry. And until then, happy studying, and I'll see you in either lecture or lab, whatever we have next.